Hello everyone, my name is Uzair Hashmi, and for my profile details, please visit sqadia.com. Now, coming towards today's lecture, we are going to discuss about the cardiac muscles, the physiology of the cardiac muscles, what are the structure and the functions of the cardiac muscles. This will be followed by the function of the heart as a pump and the heart valves. So let's begin. In this lecture, we are going to start off from the physiology of the cardiac muscles. We are going to discuss the structure of the cardiac muscles, type of the cardiac muscles, and leading towards the functions of the cardiac muscles. This will be followed by the Purkinje Fibers, and we are going to discuss its role in the action potential that will be leading ultimately towards the contraction of the heart. After that, we are going to discuss in details about the action potential of the cardiac muscle fibers, and action potential will lead towards the cardiac cycle, the excitatory cardiac muscles fibers. This would be followed by the relationship of the heart sounds to the heart pumping, and we are going to discuss in details what happens in case of the irregular heart pumping and how to distinguish all of those pumping from the sounds. We are going to discuss in details about the work output of the heart, and we are going to discuss how the heart uses the energy and from where the energy comes for the pumping of the heart. We are going to discuss in details about the chemical energy required for the cardiac contraction. This includes the atrial contraction, the ventricular contraction, majorly atrial systole and the diastole, and the ventricular diastole and the systole. And last but not the least, we are going to discuss the regulation of the heart pumping. And what I mean by the regulation of the heart pumping is by the sympathetic and the parasympathetic nervous systems that control the heart pumping. Now let's begin. So in the very first slide, this particular diagram shows the extensive structure of the heart. Now the heart is composed of three major types of the cardiac muscles as shown here. The atrial muscles shown here, the right atrial and the left atria, the ventricular muscles, these two here, the left ventricle and the right ventricle, and there are now the number three is the specialized excitatory and the conductive muscle fibers. Excitatory includes the sinoatrial node that we are going to discuss in detail in the coming slides, and the conductive includes the atrioventricular valves. Now here, if we just summarize this particular diagram very quickly, the heart basically collects the blood from the body and pumps, in summary, towards the liver for oxygenation. After this oxygenation, the blood comes back to the heart via this particular atrium, the left atrium, which contains the oxygenated blood. This oxygenated blood is then pumped down into the left ventricle. This left ventricle then contracts and sends the blood towards the whole body through aorta. Now let's move forward. We're going to discuss in detail about the physiology of the cardiac muscles. The cardiac muscles is striated, and what I mean by striated is it contains lots of striation. These dark lines followed by the light portions. And this is similar in case as compared to the skeletal muscle fibers. The cardiac muscles has a typical myofibrils that contain both the actin microfilaments and the myosin myofilaments. And these again are the identical to the structure or the physiology of the muscles or the muscle fibers that are present in the skeletal muscles. The cardiac muscle is the syncytium of many heart muscles, and what I mean by syncytium is it contains the intercalated disc, actin filaments followed by the myosin filaments, then actin, then myosin filaments. These striated portions are the intercalated discs. Now let's move forward. In this particular figure, we are going to discuss in details about the third type of the muscle fibers, that includes the excitatory and the conductive muscle fibers that we discussed in the previous slides. The Purkinje fibers. The Purkinje fibers, their location, they are basically located in the inner ventricular valves, and these inner ventricular valves, they are present just beneath the endocardium, the inner lining of the heart. This basically are the specialized myocardial fibers, and whatever excitation occurs within the heart with the help of the brain, that includes the sympathetic and the parasympathetic nervous system, is because of the Purkinje fibers. And it is because of this reason that the electrical stimulus or the impulses are enabled, which basically enables the heart to contract in a very coordinated fashion. First of all, atria contracts just prior to the ventricle contraction. The atria after contraction sends blood to the ventricle. Immediately the ventricle fills, known as the period of the diastole. After the ventricles have been filled, the atrioventricular valve will be closed. We are going to discuss in details. The ventricles will contract and the blood will move both through the aorta and towards the lung for the oxygenation and to the rest of the body. So in order to do all of these, we have the Purkinje fibers, the sinoatrial node that is present towards the right atrium, and the atrioventricular nodes. The sinoatrial node, basically, what the function of the sinoatrial node is, it basically sends the signals towards both the atriums so that the atrium contracts towards the P valve of the ECG that we are going to discuss in details. After the atriums have been contracted, the conduction signal then moves towards the atrioventricular node. The atrioventricular node is present in the conjunction between both atrium and the ventricle. The atrioventricular node then follows the His bundle. This His bundle then bifurcates and moves towards both the right atrium and the left ventricle. The left ventricle and the right ventricle. So for the contraction to occur in the ventricular systole during the QRST wave of the ECG, we're going to discuss in detail. So in summary, the Purkinje fibers, they are located in the inner ventricular walls of the heart, just beneath the endocardium. What are these? These are the specialized myocardial fibers that functions to conduct electrical stimulus that has come from the sympathetic nervous systems, and that would enable the heart to function properly and to coordinate in a very synchronous manner. This again explains the physiology of the cardiac muscles. The atria, they are separated by the ventricles. Now this slide is basically explaining a bit about the valves that are present in the heart. The atria, they are separated from the ventricles, and the thing that separates the atria from the ventricles are the fibrous tissues. These fibrous tissues are known as the valves. There are two types of valve: tricuspid valve and the bicuspid valves. Now these fibrous tissues, known as the atrioventricular valves, they are basically openings between the atria and the ventricle. Now the most important function of the valves, whether they are present between the atria and the ventricle or the ventricle and the aorta, or also known as the aortic valves, is to ensure that the blood will flow in only one direction. For example, when the atria will contract, the blood will move from the atria towards the ventricle. Here it can be seen over here. The atria after contraction will send the blood towards the ventricle, here and here, from here to here. Now in this particular slide, what is basically happening is the blood is flowing from the atria to the ventricle. When the blood has flown from the atria towards the ventricle, this particular valve, known as the atrioventricular valve, it will prevent the backward flow of the blood when the ventricles will contract, so that the blood will go only towards the aorta or towards the lungs, not back towards the atrium. Now, the division of the cardiac muscles into two functional system allows atria to contract. Now, this is very important to understand. Now, because the the heart has been divided in two major portions, the atria and the ventricle. Now these two division allows the heart, allows the atria to contract just before the contraction of the ventricle, so that when the ventricles contract, the arteria or the atrials have been contracted already, and this would result in the sending of the blood from the atrium to the ventricle, and after that the ventricle contraction will occur. Now let's discuss in details about the action potentials that are present in the cardiac muscles. Now, action potentials, as the name indicates, are the potentials, are the excitatory signals that basically triggers the contraction of the muscles. Here you can see the graph between the seconds and the action potential in the millivolts. Now here you can see that the ventricular muscle contraction begins from around minus 85 millivolts, and it causes the immediate depolarization. This will be followed by a resting phase of about 0.2 seconds. Now this 0.2 second creates a phase known as a plateau phase. After this plateau phase, when the plateau phase has been over, this causes the immediate repolarization of the ventricular muscles, where it will relax in the period known as the ventricular diastole, and this depolarization basically causes the ventricular systole, the contraction where the ventricles will contract and sends the blood both towards the lungs, which in case would be deoxygenated for the oxygenation, and the other ventricle, which will be sending blood through atria, aorta, which will be of course oxygenated. Now here, after the initial spike, the membrane remains depolarized for about 0.2 second. This 0.2 second plateau phase is not present in the skeletal muscles, and we are going to discuss in detail why is it so. This presence of the plateau phase in the action potential, this will of course cause the ventricular contraction to last as much as 15 times as that of the skeletal muscles. Like I have told you earlier, that the skeletal muscles does not contain that particular plateau phase, which is basically the prolonged contraction of the ventricular muscles. So in this particular case, what is basically happening is that the ventricles in the case of the action potentials are contracted for a longer time, 1/10 of the second or 1/100th of the second, while in case of the skeletal muscle, because the plateau phase is not present. Now if the plateau phase is not present, the depolarization will be immediately followed by the repolarization, and there will be no pause, as this pause can be seen here in the form of the plateau phase. Now this plateau phase causes the prolonged contraction of the cardiac muscles. Now this prolonged cardiac contraction allows the very efficient pumping of the heart, which is of course absent in the case of the skeletal muscles, any skeletal muscles. Now, why is this action potential of the cardiac muscles so long? It's a very valid and important question to consider, and for that, in order to answer that, we should be taking care of this particular question, that there are two major differences, which basically differentiates the action potential of the cardiac muscles from the action potentials of the skeletal muscles. Now, the action potential of the skeletal muscles, because the action potential of the skeletal muscle is fast, so we can assume that in case of the skeletal muscles, the voltage-gated sodium channels along with the potassium channels, they are present, and these are very fast channels. And they causes the excitation and the de-excitation in 1/1000th of the second, as compared to the action potential, the excitatory action potential of the cardiac muscle, which lasts for about 1/10th of the second, much longer than the excitation of the action potential of the skeletal muscle, which is about 1/1000th of the time. Now, here the most important thing is, as mentioned here, the same fast sodium channels as those in the skeletal muscles. Now the difference between the action potential of the cardiac and the skeletal muscles is in the two reasons. The cardiac muscle fiber does contain the same sodium channels that the skeletal muscles does have. Besides, the other type of the skeletal muscles which does not have the action potential of the cardiac muscles, include the entirely different population of the calcium channels. Now these calcium channels must be slow so that the excitation remains for a longer time. Now these slow calcium channels are also known as calcium sodium channels. Now, what is the importance of the calcium sodium channels in the cardiac muscles? Of course, the cardiac muscles, as I have explained previously and in this slide as well, that the cardiac muscle contracts for a much longer time. And the plateau phase of the previous slide was the exact demonstration of when it is occurring during the ventricular contraction. Now, the calcium and the sodium channels, they are slower to open. They do not open rapidly as the sodium channels or the potassium channels that open rapidly when the excitatory signal comes, causing the depolarization. Now, if the calcium channels are opening slowly along with the sodium channels, it will remain open for a much longer time. Because of the slow opening and the slow closing of the calcium channels, and after they have been open for a longer time, when they will close, as compared to the sodium channels, they will be closing much more slowly as compared to the potential sodium channels of the skeletal muscles, which will of course cause the excitation to remain much more longer as compared to the excitation that is present in the skeletal muscles. Let's move forward. Now here, the calcium channels that enter during the plateau phase. Now this particular sentence is of very prime importance. What I mean by the plateau phase, as we have discussed in the previous slide, that this plateau phase, now the calcium channel, because this particular period has been prolonged for about 0.2 seconds. So in that 0.2 second, when the calcium channels will be coming, they will of course activate the muscle contractile process. The muscle will contract much more swiftly and strongly and for a much longer time, because the calcium channels are entering. They will move from the sarcoplasmic reticulum of the T tubules. They will move and they will, attach themselves to the actin myosin complex to cause much more contraction. Also, while the calcium channels, they cause the skeletal muscles contractions, are derived from the intracellular cytoplasmic reticulum. Now this point is of very prime importance. What I mean by saying this, in case of the cardiac muscles, calcium channels, they are coming from the outside and they are moving towards the cytoplasmic reticulum and causing the contraction. Now the calcium channels of the skeletal muscles, as compared to the calcium channels of the cardiac muscles, the skeletal muscles of the skeletal muscles have the calcium channels within the sarcoplasmic reticulum, and the calcium channels are not coming from outside to the inside of the body of the muscle cell, but indeed they are coming from the sarcoplasmic reticulum that is present within the muscle cells. And they are derived from the calciums from the intracellular sarcoplasmic reticulum. So to summarize this particular diagram, the two major differences we have discussed in this particular slide, only one difference is mentioned. The other difference is present in the coming slide. We are going to discuss in detail, and that is of the permeability of the membrane. Now, one of the important difference of the action potential of the cardiac muscle is that the action potential is managed or controlled by two types of channel, calcium sodium channels in the cardiac muscles, as compared to the sodium channels in the skeletal muscles. Now the calcium sodium channels are much more slower to open, and they remain open for a longer time, and when they will close much more slowly as compared to the sodium channels, because they open rapidly, they remain open for a very small time, and they will close, and when they will close rapidly. So this causes the prolonged contraction phase of the plateau that we discussed in the previous slide. The second difference is in the permeability of the cardiac muscle for the potassium ions. The permeability of the cardiac muscles for the potassium ion decreases about 5-fold. What I mean is that from skeletal muscles, if five potassium ions are moving, only one will move from the cardiac muscle fiber, so it will decrease towards the fivefold, an effect that does not occur in the skeletal muscles. Now, why is it prime importance, which causes the action potential of the cardiac muscle to prolong for a such longer duration? Now, when the potassium ions enters, it causes the release of the potassium channels, and the repolarization occurs, which basically leads towards the closing of the the contraction period of the muscle is over whenever the potassium ions are entered into this particular muscle fibers. So the permeability decreases of the potassium ions in the case of the cardiac muscles. This gives the cardiac muscle much more prolonged time for the contraction to occur and to remain in the contraction, because the potassium ions are moving much more slowly in the cardiac muscles as compared to the potassium ions that are moving very fastly into the skeletal muscle, which will causes the repolarization and the contraction process to over. This is followed by the velocity signal conductions in the cardiac muscles. What I mean by saying this is the cardiac muscles, because the signal has been conducted, so the excitatory action potential moves with the speed of 0.3 to 0.5 meters per second. Now, this speed is of prime importance. This is about 1/250th of the nerve fiber, and this speed is 1/10 of the speed of the skeletal muscle fiber. Now let's move towards the refractory period of the cardiac muscles. Now, refractory period, as the name indicates, is the time during which a normal cardiac muscle cannot re-excitate to an already excited cardiac muscle. This means that a cardiac muscle has been excited, it has been de-excited, and there is a particular very small frame of time in which the excited muscle cannot be re-excited, because it is being de-excited very slowly. Now, at this particular peak is showing the refractory period, and refractory period of the ventricle is 0.25 to 0.3 seconds. And in case of the atria, this refractive period is much more lesser, that is 0.15 second, because the atria contains the volume of the atria is much more smaller than the atria, than the ventricle, and because the atria has to send blood in the very neighbor towards the ventricle, as compared to the ventricle, which will be sending blood away towards the lungs and towards the whole body. So the refractive period of the ventricle is much more longer than the refractive period of the atria, that is 0.15 seconds. Now, this particular figure, this particular very small p, explains the relative refractory period. Now, what is relative refractory period? The relative refractory period is a period in which the muscle is much more difficult to excite, because the refractory period here is ending. The refractory period is almost ending. So after the refractory period is over, muscle will be able to re-excite themselves. So just prior towards the ending of the refractory period is the relative refractory period, and the relative refractory period is considered within the refractory period, but this particular portion of 0.05 second makes the muscle to excite much more difficult. Nonetheless, the muscles can contract in this particular relative refractory period in case the excitatory signal that is coming is very, very strong. This will cause, of course, the excitation to occur in the relative excitatory period. For example, in case of the flight or the fight, when the heart is pumping very fastly, we are going to discuss the Frank-Starling mechanism. In those particular scenarios, we can show that the relative refractory period can be accessed to contract the muscles. This, of course, is known as can be considered in the premature contraction, because the refractory period is not over, but the contraction is still occurring. So this is not the mature contraction, because the mature contraction would have been started after the refractory period is over here. But if the contraction starts just before the refractory period, that is within that 0.05 second time span, we will consider this as the premature contraction. Now, action potential in the cardiac muscles, we are going to discuss about the excitation contraction coupling function of the calcium ions and the transverse tubules, also known as the T tubules. In case we're going to discuss in details when we're going to discuss the anatomy or the physiology of the skeletal muscles, how this whole muscle contraction occurs. Now, the focus of these two figures is primarily on the release of the calcium signals. These two diagrams explains the excitatory signals of when the calcium ions are released from the sarcoplasmic reticulum and they bind to the actin myosin head to perform the contraction. Now here in this particular figure, the axonal terminal is connected with the muscle terminal, and with the synaptic cleft, the neurotransmitters here are released. These release of the neurotransmitter will then causes the action potential to generate, and this generated action potential is prolonged along the sarcolemma and down towards the T tubules. This action potential is moving from here and moving from here, and here you can see the sarcoplasmic reticulum. When the action potential will touch the sarcoplasmic reticulum, it will trigger the release of the calcium ions. These calcium ions, after they are being released, they will bind to the troponin. We are going to discuss in detail what are the troponin, tropomyosin, and the actin and the myosin filaments in detail in the sections of the skeletal muscles. Now, after the calcium ions are being released, they will bind to the troponin and they will cause the contraction. After the contraction has been done, the calcium ions are being released by the active transport with the use of ATP, and they will move back towards the sarcoplasmic reticulum. And they will wait for the next action potential to come for the muscle to contract. That occurs both in case of the voluntary muscles and the involuntary muscles. This figure again explains, but in much more details, that the sarcoplasmic reticulum, here you can see the sarcoplasmic reticulum, the action potential will come from the axonal terminal. They will move towards, and the acetylcholine, ACH, which are the neurotransmitters, they will be released. They will bind to the sarcolemma, and after binding, they will trigger the action potential, towards the T tubules, the transverse tubules, which contains the transverse tubules then will of course are binded by the sarcoplasmic reticulum, which will then trigger the release of the calcium ions. They will bind towards the head and they will cause the contraction to occur. Now, in case of the sarcoplasmic reticulum and the T tubules of the cardiac muscles, in the previous slide we discussed the excitatory potential, excitation, and the contraction coupling function of the calcium ions in the skeletal muscles. In this particular slide, I'm going to discuss the differences and how the calcium ions and the whole process slightly differs from the cardiac muscles, which of course ultimately results in the increased contraction as seen by the plateau phase in the action potential slide previously. Now, the calcium ions that are released into the sarcoplasmic reticulum, sarcoplasm from the cisternae of the sarcoplasmic reticulum. Of course, this also occurs in the skeletal muscles, but a large quantity of the extra calcium ions also diffuses into the sarcoplasmic reticulum from the T tubules themselves at the time of the action potential. Now, this particular extra calcium is not present inside the sarcoplasmic reticulum, but it is coming from the outside of the cell towards the inside of the cell. Of course, this will result in increased calcium ion production, or presence of the calcium ion will be much more greater. When the calcium ion will be greater, they will bind to much more troponin molecules, troponin heads, and they will cause the very strong contraction. Now, the other thing that is important to consider here is the T tubules. The T tubules of the cardiac muscles. They have the diameter as much as the 5 times greater than the diameters of the T tubules of the skeletal muscle fibers. Now, what this means is that it has 25 times much more volume. than the volume of the T tubules of the skeletal muscles. This, of course, ultimately alters and enhances the contraction of the cardiac muscles. Now, this strength of the cardiac muscles, it has been established now that the strength of the cardiac muscles is dependent upon the two things. The number one is, of course, the content, extent, or the concentration of the calcium ions in the extracellular fluid, of course. Number one. Number two, the most important thing is of course, that is innate, that cannot be changed, is the diameter of the T tubules, which increases the volume for the conduction of the action potential in the cardiac muscle, which of course prolongs the contractile phase of the ventricle and hence the cardiac contraction as compared to the skeletal muscle. Now, let's begin the cardiac cycle. The cardiac events that occur from the beginning of one heartbeat towards the beginning of the next is basically the cardiac cycle, because it's a cycle, so it starts from one, ends at the other. The cardiac cycle, if we majorly divide the cardiac cycle, it will be divided into two phases. Number one is the diastole, the other one is the systole. Now, the diastole, as the name indicates, is the period of relaxation. Now, when the atria and the vesicle are relaxing, or during the phase of diastole, they will be relaxed only when the blood will be filled. This is followed by a period of systole. In this period, because in the diastole, the heart or the atria and the ventricles have already been filled. So in the systole, the ventricle and the atria will contract, resulting in the contractile phase that is known as the systoles. And I have discussed in details how these systoles and the diastoles, they are generated. They are generated by the action potential that is fired at the very top of the right atrium, known as the sinoatrial node, SA node, that then contracts the atrium. This is followed by the transmission of the conduction towards the sino-AV or the atrioventricular node. This then moves towards the bundle of His, towards the Purkinje fibers, and after the Purkinje fibers, the ventricles will contract. The sinoatrial node, of course, is the impulse generating, also known as the pacemaker. It's basically the natural pacemaker of the tissue that is located in the right atrium of the heart, that is basically towards the top of the right atrium. And these cells are basically the modified cardiocytes, are also known as the cardiac myocytes. Now, the cardiac cycles. The electrocardiogram. We are going to discuss in detail about the ECG or the electrocardiogram, also known as, sometimes also called EKG. It's basically the interpretation of the electrical activity of the heart over the period of time. Now, let's begin. Now, at this particular line, when the line starts just prior to the P phase, at this particular phase, the sinoatrial node is excited. This sinoatrial node, after excitation, moves towards the bundle that is present on the both atrium, the right atrium and the left atrium. This causes the excitation of the atria. The contraction of the atria occurs during this particular phase known as the P phase. As you can see, this phase is for the atria. This very small phase after this P phase, P peak has been eliminated, is basically the phase where the atria will relax, and after the atria will relax, the blood will flow towards the ventricular, the AV valve will close. This will be followed by the wave known as the QRS, where the ventricles are basically filled, from the blood that comes from the atria. This transitory phase is the phase where the AV node will send the electrical conduction nerves towards the bundle of His and towards the Purkinje fibers, and in this particular phase known as the T phase, the ventricles will contract. And this phase is known as the ventricular systole. This phase is known as the atrial diastole. Now the P peak is showing basically the atrial diastole, and the T is showing the the ventricular systole, the contraction of the ventricle. Now let's move forward. What we have discussed in detail, I am going to summarize here the events during the ECG. Action potential spreads from the SA nodes to the AV node, causing atrial depolarization. So as it has been established that the atrioventricular, the atrium at the top of the atrium is the sinoatrial node. It will send signal to the atrium for the atrium to contract. After the atria has been contract, the signal will move towards the AV node, causing the atrial, of course, depolarization. After this depolarization has been occur, this can be seen by the P wave, as I have explained in detail about in this particular figure. This basically induces the atrial systole. After this particular phase is of the atrial systole, where the atria will contract. The action potential, of course, will spread towards the bundle of His and the Purkinje fibers, causing the ventricular depolarization. And the ventricular depolarization is shown by the QRS complex, as shown here. In QRS phase, the peak, QRS is basically explaining the contraction of the filling of the blood, the depolarization of the ventricle. This basically induces, QRS basically induces the ventricular systole. The ventricular systole is a phase where the ventricles will contract, and they will be sending deoxygenated blood to lungs, of course, and the oxygenated blood through the aorta to whole of the body. Now, the ventricular repolarization is shown by the T wave, as explained in the previous slide here. It is shown by the T wave. Now let's discuss a bit about the cardiac cycle. Now the cardiac cycle, this particular line shown in the greenish color, bit greenish color, muddy green color, is the electrocardiogram. We are going to discuss its correlationship with the ventricular volume, with the ventricular pressure, atrial pressure, and the aortic pressure. Now let's begin. This is basically the isovolumic contraction. This particular phase is the isovolumic contraction, where the atria has been contracted and the ventricles are being filled. This phase is known as the ejection phase. By what I mean by ejection phase is, ejection means, of course, something has been ejected, and here this ejection means that the ventricles have been contracted and the blood has been ejected from the ventricles. Because the blood has been ejected from the ventricles, so the ventricles will contain less blood, so the volume of the blood will decrease, as shown here. The volume of the blood that is present in the ventricle will decrease, and this will be caused by, this is due to the ventricular systole, as shown by the T peak. Now the aortic pressure increases. Of course, the aortic pressure will increase due to the reason because the ventricle has been contracted, and the blood in this particular phase is basically moving towards the atria, and it contains a lot of blood during this particular phase passing through the atria, and the aortic valves are basically preventing the backflow of the blood from the atria towards the ventricle. Now, at the beginning of the isovolumic contraction, the AV valves are closed. AV valves are closed because the atria has been contracted and the ventricles are being filled, so the AV valves are closed so that when the ventricles will contract, blood does not backflow towards the atria. Now, after the ventricles have been contracted, this ejection phase or the aortic pressure will increase, of course, and the AV valves are open. At the end of this particular phase, the AV valves are open because the ventricles have been contracted, the volume of the ventricle has been decreased, and there is a vacuum that is being created. The vacuum that is being created in the ventricles, so the AV valves will open. Now, what will AV valves opening will do? The AV valve, when open, it will cause the entry of the blood from the atrium, both right and the left atrium, into the ventricles. The AV valves opening will cause, of course, the influx of the blood, and this will increase the volume, as shown here, that the blood volume increases from 50 ml and move towards the end of around 110 ml into the ventricles. This particular diastole will occur at this particular phase where the atrial pressure will increase, as shown by the dotted line explained in the A. This atrial pressure, this increase in the atrial pressure will of course cause the pumping of the contraction of the atria. When the SA node will trigger the excitatory signals, this of course will cause the contraction of the atria, and the blood will enter into the ventricle, as shown by a bit more increase from 115 ml to around 130 ml. At this particular point at C, as can be shown here, the AV valves will close. The closing of the AV valves is basically a prevention, that when the ventricles will contract, it will of course eliminate the chances of the blood to move back towards the atria. And when the ventricles will contract, of course, the signal will move down, because the volume will be decreased, and of course aortic pressure will increase, because blood at that particular moment when the ventricles are being emptied will be moving through the aorta, causing an increase in the aortic pressure and both the ventricular pressure, as shown by the red line. Now this is basically a cartoonic diagram where we can discuss in detail of the events that have been occurred previously. Here the cardiac muscle is basically this diagram. Towards the left is explaining the diastole, the light is explaining the systole or the pumping. In the diastole, that is the heart fillings, the AV valves are being opened and the blood is entering from the atria towards the ventricle, both from the atria towards the ventricle and the atrioventricular valve, AV valves, they are being opened. At this particular point, it is important to consider that these are the semilunar valves. They are closed at this particular moment because AV valves are opened and they are causing the influx of the blood that is coming from the posterior and the inferior vena cava towards the ventricle and from the pulmonary veins into the ventricles. Now, what will happen when the systole will occur? When the systole will occur, as you can see here, the atrioventricular valves, they will be closed so that the blood will move from the ventricle towards the aorta and towards the lungs through the pulmonary artery. One of the important points that can be considered is these are the valves of the ventricles, the ventricular muscles. Now it's important to consider that this particular ventricle, the wall of this particular ventricle is much more thinner than this particular wall. This is because the muscle fibers, quantity of the muscle is very much less, because this particular ventricle has to send blood near in the proximity towards the lungs. So a small muscle quantity would be enough for the contraction. However, in this particular case, the left ventricle, because the left ventricle is been sending blood through the aorta to whole of the body, so a much more strong contraction is needed. Now let's discuss about the relationship of heart sounds to the heart pumping and the abnormalities that we can see. Now, in this particular figure, we can see the first, second, and the third heart sounds. This is followed by the atrial sounds. Now, it's important to consider in case of the normal beat, the third and the atrial sounds have a very low rumble as compared to first, second, and the fourth heart sounds. Now, most of the abnormalities occur due to the rheumatic fevers, which is basically an autoimmune diseases which causes the destruction of the valves, hence resulting in the abnormalities. The major abnormalities discussed here are the stenosis and the regurgitation. A stenosis is when a valve in which the leaflets adhere to one another so extensively that blood cannot flow normally. This is at this particular point, a valve is said to be stenosed. Stenosis can occur in the mitral and the aortic valves. And to summarize is, stenosis is that particular process when the aortic valve or the mitral valve, they are adhered to each other so much that they are not allowing the blood from one compartment of the heart towards the other compartment, from, let's say, from the atria to the ventricle and from ventricle to the aorta. This particular phase is known as stenosis, where the valves are being stuck or adhered together and they are not allowing the blood flow from one compartment of the heart towards the other compartment of the heart. The regurgitation is a condition where the edges of the valves, they are so much destroyed, and that destruction can be due to various factors. This can be due to the rheumatic fever, and can be due to the scar tissue, due to any inflammation, endocarditis, due to any other complication, that the walls of the ventricles, they are not closed, they are not contracted properly. And this causes, because the function of the valve is died when the valves are not closing properly, and this can occur due to various reasons. One of the reasons that has been discussed is the development of the scar tissue, which will prevent the closing of the valve. Now, when the valve will stop closing after the blood has entered from one compartment to another compartment, this will cause the backflow of the blood. This backflow of the blood is known as the regurgitation, and this regurgitation can occur wherever the valves are present in the aorta. This can occur in the mitral valve, this can occur in the aortic valves. Up till now, we have discussed the physiology of the cardiac muscles, the Purkinje fibers. We have discussed the action potential of the cardiac muscles and how the action potential of the cardiac muscles is different from the action potential of the skeletal muscles. We have discussed the cardiac cycles. We have discussed the diastole and the systole, its relationship with the ventricular volume, its relationship with the atrial and the ventricle pressure, and the aortic pressure and the aortic volume. We have also discussed about the relationship of heart sounds to heart pumping. Now we are going to discuss the work output of the heart. Now, the stroke work output of the heart is what? It's basically the amount of energy that is needed by the heart and the energy that is converted by the heart to work during each single heartbeat when the blood is being pumped from the heart towards the body. Now, the major portion of the work output is used to move blood from the low pressure veins towards the high pressure arteries, that is from the inferior and the superior vena cava, and from the lungs towards the arteries. The second and the most important function of the work output is that a very small but accountable proportion of the energy is used to accelerate the blood through the aortic and the pulmonary valves. Now, let's summarize the two important functions of the work output of the heart. Number one is it functions, the major proportion of the energy that is being produced during the work output is used to move the blood from the low pressure veins towards the high pressure arteries. After the blood has been moved from the low pressure vein towards the high pressure arteries, because the blood is in the very slow moving form, the rest of the energy, a minor proportion of the energy, is used to accelerate the blood through the aortic and the pulmonary valves. These are very important because if at this particular point the blood is not accelerated, there is a very large chance that the blood will cause the regurgitation or the backflow will occur in case there is a very minor ambiguity, there is a very minor dysfunction of the valves as discussed previously in case of the regurgitation or the stenosis. Now, this particular diagram explains the graphical analysis of the ventricular pumping. Now, it's being divided into four phases. Phase 1 is known as the period of filling, where the blood starts filling, because this is the ventricular pumping. So we are going to discuss specifically about the ventricles. The blood starts filling from 45 ml and it moves towards 115 ml during this particular phase, known as the period of filling. The second phase, known as the period of isovolumic contraction, is the phase during which the diastolic pressure will cause the increase in the volume from 115 to about 250, a bit less than 250. Along with that, the pressure, the intraventricular pressure will increase. That the increase in the intraventricular pressure is because of the conduction of the nerve impulses from SA to AV node, and from AV to bundle of His, and bundle of His to Purkinje Fibers, where the cardiac muscle starts to contract. But here in the case of the diastolic pressure, the ventriculars are filled to the maximum extent. Phase 3 is the phase of ejection. This is also known as the phase of ejection. And why is known as the phase of ejection? Of course, because the ventricles have been filled during the diastolic pressure, and during this phase, the phase 3, the ventricles will contract because the excitatory signals has reached towards the Purkinje fibers, and they will cause the contraction of the cardiac ventricles, or the ventricular muscles of the cardiac fiber of the cardiac. This causes the systolic pressure. The systolic pressure that is represented by the T peak of the PQRST wave that we have discussed in the diagram is explained by the systolic pressure, where the ventricles will contract, the blood will flow from the ventricles through the aortic and the pulmonary walls to the aorta, and from aorta to the rest of the body, and from the pulmonary walls towards the lungs for the oxygenation. After this period of ejection, or the systolic pressure, is over, this is followed by the period of isovolumic relaxation. The period of isovolumic relaxation is when the ventricles have, after contraction, are being relaxed. This is a very small phase, resulting in the immediate depression of the volume of the blood from about 120 to directly 45 ml. Now let's talk about the chemical energy required for the cardiac contraction. The energy, of course, is derived mainly from the oxidative metabolism of fatty acids, number one, and to a lesser extent the other nutrients are also included, which includes especially the lactate and the glucose. And one of the important mechanisms that we are going to discuss in the later lectures is of the flank Starling mechanism. Now, what flank Starling mechanism is? It's basically the ability of heart to adjust towards the increasing volumes of the blood that is being inflowing from the body towards the heart. Now, what I mean by saying this is that the flank Starling mechanism allows the heart to adapt or to accommodate the greater volume of the blood that is being entered in towards the atrium, and from atrium to the ventricular towards the aorta and towards the whole body. And this mechanism is of great importance, and we are going to discuss in detail about the flank Starling mechanisms in the coming lectures, but I thought it is important to mention the flank Starling mechanism at this particular point. Now, talking about the efficiency of the cardiac contraction, it's basically the summary during the, and we have discussed in details that during the heart contraction, when the muscles of the heart or the cardiac myocytes, they are being contracted, the chemical energy that is being converted, the chemical energy is converted into two things: the heat, which is basically, of course, the major portion, the heat. Also the small portion is converted into the work output, and the work output we have discussed in details in the previous slide, that the stroke work output is used, number one, to move the blood from the low pressure veins towards the high pressure arteries, and after the and also to accelerate the flow of the blood from the aortic and the pulmonary walls. So beside these, the chemical energy that is being converted is utilized in heat in the form of resistance. The ratio of the output to the total energy expenditure, of course, is known as the efficiency of the cardiac contraction, or simply the efficiency of the heart. Now, it's important to consider that not 100% of the chemical energy that is being converted is utilized in the work output, but maximum efficiency of the heart, of the normal heart, that is seen is to be between 20 to 25%. What does it mean? It means that 20 to 25% of the chemical energy that is available is used for the work output. The rest, 80 to 75 to 80% of the energy is being wasted in the form of heat. In case of a heart failure or the myocardial infarction, this max energy or the efficiency output or the efficiency of the cardiac contraction is reduced from 20 to 25% to 5 to 10%, which decreases the contraction process of the heart, resulting in the heart failure or myocardial infarction. Now let's talk about the regulation of the heart pumping. Now, of course, one of the very best method to express the ability or the functional ability of the ventricles to pump blood is by determining the curves of the ventricular function. Now here you can see that the atrial pressure of each side of the heart, when it increases, it causes the increase in the stroke work output until it reaches the pumping ability, the ventricular pumping ability. At this particular case, as you increase the atrial pressure, the ventricular stroke output will not increase because it has reached the maximum potential of its pumping ability. Let's discuss about the regulation of the heart excitation, and in this particular slide we are going to discuss the sympathetic nerves. The pumping effectiveness of the heart is also controlled by both of the sections, and the sympathetic nervous system and the parasympathetic nervous system. Here you can see the sympathetic veins, and these are the vagus veins of the parasympathetic system, both sympathetic and the parasympathetic systems that control the heart excitation. Here you can see these chains, both sympathetic chains and the sympathetic nerves that are ending towards near the sinoatrial node and the atrioventricular nodes. Now it's important to consider that whenever in the presence of the strong sympathetic stimulation, the heart rate of the normal person, which is about 70 beats per minute, can increase up to 180 beats per minute, 180 to 200 beats per minute, and very rarely it can increase to 250 beats per minute, and that is because of the sympathetic stimulation. So the most important function of the sympathetic system is that about 80 to 90% of the heartbeat is because of the sympathetic stimulation of the cardiac myocytes. As it increases the heartbeat that from 70 to 180 to 200, and also at sometimes towards the 250, it also increases the force of heart contraction. The force of the heart contraction is increased because of the many reasons. One of the reasons is the Frank-Starling mechanism. We are going to discuss in detail and how much the force can be increased by the sympathetic stimulation. It can increase up to 2 folds. It can double the force of the contraction. What would be the result of this particular sympathetic stimulation when the force of the heart contraction will be doubled? It of course helps in pumping the greater volume of the blood that is being ejected from the ventricular contraction, and it would help in maintaining the ejection pressure, of course, as well. Now in this particular slide, the parasympathetic stimulation of heart is mentioned, that the parasympathetic stimulation occurs to about can pump about from 0 to about a maximum of 7 ml liters per minute of the blood. in the absence of the zero sympathetic stimulation. In case of the normal sympathetic stimulation, when the sympathetic stimulation is normal, the heart output, the cardiac heart output would be between 10 to 15 liters per minute of the blood, around 13.5 liters of the blood will be pumped per minute. In case of the maximum sympathetic stimulation that we discussed, that the maximum sympathetic stimulation can can increase the heartbeat from 70 to 180 to 200%, and even sometimes to 250 beats per minute. At this particular phase, because the stimulation from the sympathetic nervous system is maximum, it will cause the flow of blood to about 23 to 24 liters per minute. Now let's discuss what we have discussed in details about this particular lecture in details. Let's go through it. The summary: we have discussed the physiology of the cardiac muscles. The physiology of the cardiac muscles include, we have discussed three types of the muscles: actin, myosin, and the conductive fibers. We have discussed the functions of them. We have discussed the Purkinje fibers, the heart valves, the sinoatrial node. We have discussed the Purkinje fibers. We have discussed bundle of His. We have discussed how the excitatory signals are being generated from the sinoatrial nodes and stimulating the atria to contract and moving towards the atrioventricular nodes, where it moves from the bundle of His to the Purkinje fibers and causes the contraction of the ventricular muscles. We have discussed the cardiac cycle. We have discussed how the cardiac cycle is basically divided into two parts, the diastole and the systole. We have discussed the PQRST wave, which basically explains the atrial systole, atrial diastole, ventricular diastole, and the ventricular systoles representing by the T wave. We have discussed the relationship of heart sounds to heart pumping. We have discussed how the third and the atrial heart sound has a very low rumble and it changes in case of the abnormalities. We have discussed how the rheumatic fevers, which is basically an autoimmune disease, can result in the destruction of the heart pumping, where you can see we have seen different types of the abnormalities. For example, we have seen atrial stenosis, atrial regurgitation, mitral stenosis, mitral regurgitations. We have discussed the stenosis and the regurgitation, where it's in the case of stenosis, the heart valves are attached so much that they prevent the flow of the blood. While conversely, in contrast, the regurgitation, the walls are damaged to such an extent by a scar tissue that they are not open, they are not able to close properly. This causes the backflow of the blood. We have discussed the output of the heart, the work output of the heart. In that particular scenario, we have discussed that the chemical energy is converted into two things: the heat in form of resistance and the work output. This work output is then used to trigger two things: to move the blood from the low pressure veins towards the high pressure arteries, and to accelerate the blood from the aorta and the pulmonary valves, so that the blood can be pumped effectively. And we have discussed the maximum efficiency of the work output is about 20 to 25%. In case of the heart failure or the myocardial infarction, it decreases to 5 to 10%. We discussed the chemical energy required for the cardiac contraction, and we have discussed the regulation of the heart pumping. That was all about this particular lecture. Thank you for watching sqadia.com. Keep watching.